so I don't forget. All right, let me get started here. I want to start off by talking about the exam. Your exam will be online. It's 50 multiple choice questions. You will have an hour and 15 minutes to complete the exam. That roughly comes out to 1.5 minutes per question that you have time to answer. This is a test. It's not an open note test. Okay, so make sure you are studying so you can answer these questions. The way it works, though, is that there's always the possibility that Blackboard will crash on you. If it does, just log back in, go back to the test, and the test time will actually should have stopped, and then it'll let you just pick up from where you left off. So if you happen to get booted out, don't freak out. Just get back into the system and continue taking the test. Outside of that, based on all the faculty in our department, if there are any issues with students running out of time, it's because they didn't study for the test and they're trying to treat it as an open notes test. So please understand, as far as excuses go for not being able to complete it, it's going to be real hard to come up with one that doesn't result in you still getting a 0. So please just be prepared. Treat it like a regular exam. Study like crazy for it. Okay? And again, the exam will begin at 9 a.m. When you log into Blackboard, you'll look at the icons. There's going to be a new icon and it's going to say exams. Click on that folder or that icon that says exams. And in that folder will be your test, which you will click on to start your test at 9 a.m. Again, 9 a.m. to 10:15. If you are an SAS student, when I set up the test parameters such as time and the date, I'm able to select specific students that are SA students that need extended time, and that's where I add on time for those students, but only those students that have sent me the accommodation form. Okay? Are there any questions beyond that? Okay. Another thing is I will be, by my computer. What I'm going to do is go ahead and pre-record a lecture on Chapter 7 because Chapter 7, with the time that'll be left in class, it won't be enough time to cover that whole chapter, and I would like to lecture on the entire chapter as one. So I will pre-record the lecture and I'll post the announcement on Blackboard when I've completed it, along with Lab 4. I'll pre-record Lab 4 also. But for the lecture, this is at one time. What we call it, lectures will usually be live outside of that. All right, let's go ahead and get going here. We were talking about the 4 types of organic compounds here in Chapter 5. We talked about how organic compounds, first and foremost, what they have in common is that they're all carbon-based. In addition to carbon, you find a lot of hydrogen and either a lot or a little bit of oxygen contributing to their formations. And if it's in the case of proteins and nucleic acids, you'll also find nitrogen contributing. Now, all these atoms, what they have in common is they're unstable atoms. Their valence shells do not contain the maximum number of electrons, and therefore they try to become more stable by reacting with other unstable atoms. Now, in the case of organic compounds, when these unstable atoms react with each other, centered around carbon, primarily what results is the formation of covalent bonds. So these are very stable compounds regardless of the surrounding environment. Now, we already discussed carbohydrates, we discussed proteins, and we discussed nucleic acids. And we talked about how these 3 out of 4 organic compounds, when you look at their most complex forms, it was discovered that they're made up of smaller units or smaller molecules that have similar structures that we call monomers, such as for the case of carbohydrates, the carbohydrate monomer was a monosaccharide. And if you take a monosaccharide such as glucose and covalently bond it to many other molecules of glucose, we end up creating a polymer or a carbohydrate polymer that we call a polysaccharide, such as glycogen or starch or cellulose. And basically it followed that same pattern as you moved into proteins and as you moved into nucleic acids. But as we also talked about, that's the basics for those 3 when you talk about monomers and polymers. Because as I discussed for proteins and for nucleic acids, when we get to the level of the polymer, okay, it doesn't always end there. Such as in the case of proteins, we discussed 4 levels of protein structure: primary, secondary, tertiary, and quaternary. And at the quaternary level, that is a very complex protein made up of multiple polymers called polypeptides. We talked about nucleic acids, and we talked about nucleic acid polymers known as polynucleotides. But when it comes to DNA, DNA is not made up of one polynucleotide, as we discussed. It's made up of two. Okay, but there is somewhat of a pattern there. Now, the fourth organic compound that we left off at, they're called lipids. Okay, lipids are famous for being hydrophobic, just flat out, water hating. However, one of these lipids that we are going to discuss is going to be very important to us throughout the semester. It's called a phospholipid. Okay, however, this lipid up here, which is one of four types called a fatty acid, we need that lipid to form these other two types. Okay, and then we'll just talk about cholesterol when we get there. Okay, so for lipids, we don't talk about monopolymers. Instead, I prefer to approach them as four types, beginning with the simplest type, which is the fatty acids. Fatty acids are described as hydrocarbons. If you recall, we went over this in chapter 4. Okay, molecules that are described as hydrocarbons, they're made up primarily of carbon, covalently bonded together, forming a skeleton or backbone. And then this carbon backbone, in turn, is covalently bonded to many atoms of hydrogen surrounding it. Thus, we call this a hydrocarbon. However, a fatty acid is a type of organic compound, a lipid. On one end of this, we have our functional group. If you recall, this is one of 7 functional groups called a carboxyl group. So we do have a little bit of oxygen. Keep in mind what I said before. Carbon loves to form nonpolar covalent bonds, single or double. It loves stable relationships. Hydrogen, it depends on which atom it is interacting with. If it's carbon, it's going to form a stable relationship because carbon's going to let it know this is how it's going to be. But if it's oxygen, okay, such as over here, they'll form a stable relationship. They'll form a covalent bond, but more than likely it's going to be a little what? Polar, if you remember polar covalent bond. Whenever you see oxygen, I said think of oxygen, I like to describe oxygen as a love 'em or leave 'em atom. It loves to get into relationships. It loves to react with other atoms, but if the environment is conducive to it, it'll take off and it'll even take an electron from another atom it was initially sharing with. So what I'm getting at is whenever you look at an organic compound, you not only want to see wherever there's a functional group at, but notice often, not always, but often you'll find oxygen there. Oxygen is a highly reactive atom. It's what we call an electronegative atom. So the way it works for this fatty acid here, I said lipids in general are hydrophobic, water hating. Polar water molecules are excellent at breaking ionic bonds. And I say this in the Lab 3 overview, which I pre-recorded. So there won't be a live overview of the lab. It's already been pre-recorded, should be available. And in that, and in the lab itself, it discussed or briefly talks about how any compound formed through an ionic bond, polar water molecules can break that bond easily. Okay. However, when it comes to covalent bonds, polar water molecules rarely, if ever, can break these bonds. Therefore, when you surround this organic compound, this lipid called a fatty acid, okay, when you put it in a water-based environment, the polar water molecules cannot break these bonds. Therefore, this, okay, is never dissolved or broken down, okay, or disassociates in water on this end. However, here, if water molecules were going to react, or if a chemical reaction were going to occur, such as dehydration synthesis, this is where it's going to happen, where we have the atoms of oxygen. So there's one end of this molecule, okay, that doesn't mind being by water. However, overall, we describe it as hydrophobic. Now, keep in mind, when we went over carbohydrates, the 3 key atoms you always see are carbon, hydrogen, and oxygen. And when we talked about carbohydrate monomers called monosaccharides, I said the molecular formula is always the same, C6H12O6. Now, there were only 2 types, glucose and fructose, you needed to know, but they had the same molecular formula, C6H12O6. Notice this here. There are many different types of fatty acids. This is one of the smaller ones, or if you want to call it, shorter ones. Look at its molecular formula. It's known as lauric acid. This is a fatty acid. Look at the molecular formula. C12 has many more atoms of carbon than a monosaccharide. H24, twice as many hydrogen. But notice the number of oxygen atoms. In a carbohydrate monomer called a monosaccharide, there are 6 atoms of oxygen. Notice this for the fatty acid. Even though it is much larger in structure, made up of many more carbon and hydrogen, and I said this is actually one of the shorter fatty acids, it only has 2 atoms of oxygen. So for lipids, you see a lot of carbon, hydrogen, and oxygen, but very little oxygen often. Okay, so this is lauric acid. And then you may have heard talk in regards to diet, saturated versus unsaturated fats. Well, in biochem, when we say saturated versus unsaturated fats, we're referring to fatty acids that either the carbon backbone is formed by many atoms of carbon that have formed nonpolar single covalent bonds. But if we use the term unsaturated, we're now referring to a fatty acid that again has a carbon backbone, except two of the atoms of carbon don't have a single bond chemically attaching them together, but instead have formed a double bond. And we'll talk about this in chapter 7, how this can actually change the overall shape of the fatty acid, causing it to go from having a linear shape to, if you look here, a bend in it. So notice here the difference. They're showing you how it affects the carbon backbone. Well, remember we talked about in chapter 4, if organic compounds are all carbon-based and all have a carbon backbone, why do we divide them into 4 types? We talked about how it depends on how these carbons, how many of them form bonds with each other, whether they're single or double, and what other atoms they're forming chemical bonds with. So in other words, the shape, and then the shape contributes to the function. The second type of lipid is a triglyceride. Triglycerides in lay terms are known as fats and oil. Okay, so again, the fourth type of organic compound is not fats and oil. The fourth type of organic compound are called lipids. Lipids are divided into 4 types, and the second type, they are technically known as triglycerides, but in lay terms, we refer to them as fats. In other words, stored form of fat in your body. We also refer to these triglycerides as oils, such as vegetable oil. Okay, well, in our body, when you eat a diet, okay, containing fatty acids, what'll happen is eventually these will be absorbed, some of them. They'll be transferred to cells called adipocytes, which in lay terms are called fat cells. And what these adipocytes will do is they will take the fatty acid on the end where you find the carboxyl group, okay, and this adipocyte or fat cell will take an alcohol known as glycerol, okay. You can see here glycerol has 3 functional groups. All of them are the same. They're all hydroxyl groups attached to this carbon backbone here. Okay. Now, if you look here for the fatty acid, what will happen? This is the only area, okay, that it was found that a chemical reaction will easily occur. It's not the only area a chemical reaction can occur, but easily occur. And what will happen is the adipocyte will take these fatty acids, and on this side where you have the carboxyl group, it'll perform a chemical reaction that we talked about called dehydration synthesis. Dehydration synthesis is whereby proteins called enzymes form covalent bonds to create a larger compound. But when the covalent bond is created, there are certain atoms that have to be moved out of the way to free up electrons between here and here. Well, those atoms work out nicely. The atoms that are moved out of the way to free up electrons between these 2 molecules happen to be combined together, forming H2O. Thus, we call it dehydration. The synthesis is we create a covalent bond. And what the fat cell or the adipocyte will do is it'll take 3 fatty acids, each fatty acid at its carboxyl end, and it'll covalently bond them to each of these hydroxyl groups on the glycerol molecule through that same chemical reaction, dehydration synthesis. This results in a fat molecule, okay, or oil, but here it's showing you a fat molecule. Stored form of fat is what we're talking about, such as in our body, in a fat cell called an adipocyte. And we call this molecule, once it's formed and completed, you can see here they're highlighting the areas where dehydration synthesis occurred and where the covalent bonds are formed. We now call this molecule a a tri for 3 fatty acids, glyceride for the alcohol glycerol that they're attached to. Okay, so again, I may ask you, a triglyceride is formed by what molecules? You would simply say 3 fatty acids and 1 molecule of glycerol, okay, in chemical structure. This is our stored form of fat. Okay, now this lipid here, the third type of lipid, called a phospholipid. You're going to hear a whole lot about this lipid going into chapter 6, and then after the first exam, you'll hear a whole lot more about it in regards to chapter 7. Okay. A phospholipid is the major organic compound that forms the membranes of cells. of all living organisms, okay? So whether you're talking prokaryotes or eukaryotes, you will find phospholipids forming a structure known as a cell or plasma membrane. You'll hear me use the two terms interchangeably, okay? Phospholipids are unique in that in actuality, they are both hydrophobic and hydrophilic, okay? They're the unique ones, okay? For the majority of lipids, such as fatty acids and triglycerides and our last group, steroids, okay, they're hydrophobic, highly hydrophobic, don't like to react with water, but phospholipids are unique. There's one side of the phospholipid that acts like a lipid. It hates to interact with water, but the other side of it has no problem with water, and this is important, as we'll talk about as we progress and talk about the cell membrane. Up here, they're showing you. These are three different ways to represent a phospholipid, starting with this way here, okay? The chemical structure of a phospholipid. It consists of a backbone made up of that alcohol glycerol. Just like with triglyceride, phospholipids have a backbone made up of glycerol, okay? Phospholipid also has fatty acids contributing to its formation, okay? But it has two fatty acids. There's one fatty acid here, and there's a second fatty acid here. So it's not like a triglyceride in that sense, because triglycerides have three fatty acids. A phospholipid only has two fatty acids, but they are covalently bonded to that molecule of glycerol. Instead of a third fatty acid, we instead have the functional group called a phosphate group. If you recall, I said we would use an uppercase P with a circle rather than drawing this structure out in detail. This is our phosphate group we talked about in chapter 4. Okay, so instead of a third fatty acid being attached to this molecule of glycerol, we instead have a functional group called the phosphate group. And then attached to the phosphate group here, it says choline. Okay, I'm going to try to loop this over. Instead of choline, I want you to know it simply as a, okay, somebody just joined the session and blocked my screen here. All right. Sorry, this collaborated something else. When somebody comes on, sorry for my writing here. Should say polar. Okay. Instead of saying choline, just know this as a polar group. Look in your notes. I have it written much better. I'm sorry if there's a lot of noise in the background. Let me know, and I can move. Have some little ones that are not satisfied with this morning's event. So, but if you look here, it says choline, but what I want you to know it as, and it's down here, what I want you to know it as is not choline. Know it more general as a polar group. You take an advanced cell and molecular course, and they will tell you for a phospholipid, there's not one type of phospholipid. There are different types of phospholipids, and the differences are in the polar group. Some phospholipids have a molecule called choline. Some have other molecules such as inositol and what have not. I don't want you to know that. I just want you to know, okay, this part here. So again, I may ask you, what are the smaller molecules that form a phospholipid? You would simply tell me of one molecule of glycerol plus two fatty acids plus a phosphate group plus a polar group. Okay. So this is one way of drawing it out. This is an uglier way of drawing it out. And then this way. Okay, some people call it a ball, would call it a head and tails. Sometimes I call it a ball and chain. But if you look here, more often than not, it's simply referred to as the head portion of the phospholipid and the tail portion of the phospholipid. And that's what they've done over here. They've expanded it. You will see this a lot for phospholipids, such as right here. You can see here they're showing you many of these phospholipid molecules. Now, here, the tail part. The tail part is in reference to the fatty acids. Okay, the fatty acids are hydrophobic. Now it says fatty acid tails, but just know them as fatty acids. Each of these represents a fatty acid here. They're hydrophobic. They're water hating. Up here, this little head or the little ball here, okay, it represents the other three types of molecules that form a phospholipid beginning here. Where the tails connect to this little head part or ball part, that's roughly where you would have your molecule of glycerol, right in here. Okay. Then the middle part here, that would roughly be where you have your phosphate group. And then the top part here, well, that would be your, sorry, trying to draw a little arrow. That would be the area where you have your polar group, okay, which is hydrophilic. And technically speaking, both the polar group and the phosphate group are hydrophilic, but for the test, just think of it as the polar group. This end, the hydrophilic part, is water-loving. Hydrophobic, if you recall, I said just simply define it as water-hating, okay? Because of the design, because of the smaller molecules that form this organic compound, this type of lipid called a phospholipid, what scientists discovered is that the cell membrane, also known as the plasma membrane of the cells of living organisms, okay, these phospholipids were found to form, and I'm going to erase all this stuff, were found to form a bilayer, what we call a phospholipid, oh my God, that's mad, what we call a phospholipid bilayer, okay. The bilayer comes from the fact that you have many phospholipids that form one layer, represented, which I bracketed, represented here. You can see these little guy here, which represents a phospholipid. They're showing you that the cell or plasma membrane, okay, of living cells, okay, these phospholipids actually form a bilayer. You can see many of these phospholipids form not one layer, though. They actually form two layers. Thus the bilayer part here, okay, but it's all made up of phospholipids. Now, what you can't see here, all right, is the fact that, and I'll go over this more in chapter 7, is the fact that this phospholipid bilayer, okay, would have a spherical shape to it, a three-dimensional spherical shape, or you can say three-dimensional circle or ball shape to it, okay? It would be enclosed. Now, the way it works is you have to keep, we're going to talk about this especially in chapter 7, outside of the cell membrane is a water-based solution, and I'm just going to put water. Such as the cells that form our body. If you recall, we talked about it in chapter 3. We're made up of 3/3 water. Oh my God, that was bad. We're made up of 2/3 water. Misspoke, 2/3 water. Most of that water is found within our cells. However, you find that water outside of your cells. And we're going to talk about this later in chapter 6. Okay, you have solution inside of your cell that is a water-based solution that you're going to come to know as a cytosol or ICF, intracellular fluid. Outside of our cells, we're going to talk about a little bit in chapter 6 and a lot in chapter 7. You have another solution. Okay, it's in the extracellular environment. You're going to come to know it as extracellular fluid or ECF. Okay, therefore, what they discovered is these phospholipids, they're the major organic compound that forms the cell or plasma membrane. And they're arranged in a way that they create a barrier, which you're going to come to know in chapter 7, especially a hydrophobic barrier, a water-hating barrier. Oh my gosh, I can't spell barrier at all with this thing. I thought I was getting better. Spoke too soon on Tuesday. Okay, so we have a barrier. Notice something. When these two layers are formed by all these phospholipids, notice the hydrophobic fatty acids actually come together facing each other here. What are they doing? Well, they hate water. They're turning away from water. So by having these phospholipids form two layers, this allows the fatty acids to turn and face each other, creating not only a barrier, but a hydrophobic barrier. The rest of the phospholipids, the head part that contains the hydrophilic portion, well, that hydrophilic portion there, it's facing water, which it has no problem with because it loves water. So these are very unique, okay, lipids. Okay, but the fatty acids up here, the one good thing about this figure up here is that it shows you the majority of the molecule is actually made up of fatty acids. A good chunk of it is, okay? So we put it under lipids because it does have the characteristics of lipids, hydrophobic. We'll talk about this more again in chapter 6 and 7. This is just showing you what I just drew out. I forgot I had it on here. I usually go over my PowerPoints, but this is a neater drawing than what I drew. Now you can look at that there. The last lipids. The last group of lipids are steroids. Okay. Steroids have a characteristic structural shape. It's this part right here. So whenever if you move on and you're taking classes or they start talking about steroids, okay, and they happen to give you figures of steroids before they start lecturing, if you see any molecules, figures of molecules that look like this, automatically tell yourself, okay, we're probably going to be talking about steroids then, because that's their hallmark structure. If you remember last class, I said there are so many atoms of carbon contributing to forming our organic compounds that scientists decided we're not going to keep, okay, showing you where these atoms of carbons are. Sometimes we will, such as up here where we have our functional groups called methyl groups, here, CH3. But often they'll say, you know, there's so much carbon, such as in the lipid steroids. Instead of writing out the C, everywhere you see a little sharp turn or corner, let me know if it gets too loud, everybody, and I'll move. Everywhere you see one of these spots here where I'm drawing an arrow on a few of these, not all of them. Everywhere you see one of these sharp corners or bends, okay, that lets you know there's an atom of carbon there. Okay, so there is a lot of carbon, but the carbon is not forming a linear backbone. Most of the atoms of carbon are forming a cyclical or circular backbone, or what we call a benzene ring. One type of steroid is cholesterol. Okay, cholesterol is described, okay, two ways. In regards to steroids, it's described as a precursor used by cells of an animal's body to make other steroids, such as testosterone and such as estrogen. Okay. How in this class, we're mainly going to talk about how cholesterol also contributes to forming the cell membrane. Okay. So cholesterol is both a precursor. Precursor just means, in this case, a starter molecule that is modified, okay, structurally changed to where it becomes a different type of compound with different functions. Check my spelling. Hopefully I remember how to spell. I don't know, getting older. Should say precursor. Again, I screwed that up. Sorry, I'm on a pad and it slid. I guess I'm using excuses. So cholesterol here in this case, on this figure, it's a precursor. There are cells of our body, such as in the testes in males and ovaries in females, that will take cholesterol, and there are protein enzymes which will modify and tweak this, converting it either to testosterone or estrogen. But notice something again. You still have that four-ring structure in the steroid testosterone. You still have that four-ring structure in the steroid estrogen, just like you had in cholesterol. This is a very large molecule. So if you like to cook, but you got a big family to cook for, you know, sometimes it's better to go to the grocery store and buy those pre-prepared meals. I don't want to make spaghetti sauce from scratch. Let me go ahead and just buy it out of the can. I don't want to make pasta from scratch. I'm going to buy dry noodles and boil those, that kind of deal. Well, that's what your cells are doing. They're taking this ingredient and just tweaking it here. Now, the second function of, however, of cholesterol has to do with what we call our cell membrane or plasma membrane. Okay. It also contributes to forming our cell membrane, but to the lesser degree. I'm not going to write out the rest that's supposed to be membrane because obviously I feel like I'm doing worse now. So these are two functions of cholesterol, okay, precursor. Now, you often hear bad news about cholesterol, but in actuality, it's getting a bad rap, okay? Are there any questions here for chapter 5? I thought I was really getting better with this writing, and it's horrible right now. Okay, let's move on to chapter 6. All right, for chapter 6, again, let me know if you hear a lot of that background in the noise right now. I've got a little toddler running around here, and she's the boss, so, okay. If you look here for chapter 6 now, we're finally moving up. We're now moving out of the chemical molecular level, and we are now moving to the cellular level. However, in chapter 6, while we're at the cellular level, nested underneath or included in this chapter is the organelle level. Okay. So I'll start off with some general information about cells, including talking about prokaryotes versus eukaryotes, and slowly we will transition over to specifically eukaryotes. Okay, and when we get into eukaryotes, Okay, that's when we'll go into detail about organelles. I'll mention organelles, especially between prokaryotes in comparison to eukaryotes. But it's when we get into eukaryotes that we're going to go focus specifically on talking about how organelles are divided into two groups: membranous organelles and non-membranous organelles. And we're going to go over types of each group as we go through this. And then some of these organelles you will see, okay, again later in the semester once or more. Now, Chapter 5, you need Chapter 5 and a little bit of that information I gave you on biological hierarchy in Chapter 1 to help you out with Chapter 6. Okay, so even though we're out of the chemical molecular level, if you recall, I said this course builds on itself. So Chapter 5 feeds into Chapter 6. Now, as we go through Chapter 6, the main compounds that we're going to end up focusing on as we go through Chapter 6 and into Chapter 7 will end up being eventually proteins and lipids, but we'll mention all of them as we go through this chapter. But these two here are going to be the ones we'll place a heavy emphasis on by the time we're reaching the end of this chapter, and definitely in Chapter 7. Okay. Now, cells. The study of cells is actually called cytology. Cyto. You're going to see this a lot in a bit. I'm going to jump around in your notes in a bit. You may have heard the terms cytoplasm, cytosol, things such as that. Well, we're talking about cytology. We'll also talk about cytoskeleton. The cyto means cell. Logy means study of. So cytology is the study of cells. However, in colleges, they often refer to it as cellular biology or cellular molecular biology. Go figure. Now, there is a concept, a belief in reference to cells, and they call this the cell theory. The cell theory starts off by stating, okay, that cells are the smallest basic units of life. Listen carefully. When I was an undergrad, okay, biology majors and even pre-med and nursing majors were not required to take cell and molecular biology. It was an elective. It was not one of your core courses, okay? Biochem, genetics, microbiology, anatomy, physiology, to name a few courses, those were considered much, much more important. But at the same time, I was an undergrad, cancer research really took off. I mean, cancer centers started to pop up, like the CTRC in San Antonio. And basically, you had those group of scientists that were doing research in cancer that finally told everybody else, look, we've all known about cells. We've even been isolating cells from different species, including humans. We need to finally place an emphasis on the importance of cells. So a couple of decades or so later, when I came back to teach at this same college, they had revamped their core courses. Now, not only was cell and molecular biology a required core course at this college, it was the final core course that the students would take before they would move on to grad school, med school, nursing school, or whatever job. So eventually scientists that were doing research said they're important. Well, we say that cells are the smallest basic units of life because the simplest known living organisms are unicellular. The most complex living organisms are made up of many cells. So we do not reach a level in the biological hierarchy where we can start talking about living organisms until we reach the cellular level. Okay, there have been scientists that have taken, say, a eukaryotic cell and pulled it apart into its various organelles, and nothing happens except the organelles eventually degrade. Okay, however, when you put them all together, forming that functional cell, whether it's prokaryote or eukaryote, the living organism not only will survive if you put it in the ideal conditions, the cells, okay, I'm sorry, will not only survive, but the cells will also divide. Okay, so the second component to this cell theory backing up basic units of life is cells are produced by division of pre-existing cells. We will visit cell division in chapters 12 and 13. So the way it works is over here. This dates back, the earliest known I've seen it was dated back to the 60s. When I was a grad student and a little after, I worked with what we call cell lines. Okay, these cell lines were cells that had been isolated from, in this case, people, individuals, humans, and transferred into a culture dish or a plastic vessel, if you want to call it in lay terms. And what happened is they gave these cells, okay, an ideal environment. a water-based solution with the necessary nutrients, minerals, made sure they had the gas they needed, O2, made sure they had the right pH and the right temp. And what they found out is not only would these cells survive, but these cells would begin to divide and grow to where eventually they would cover up the surface they were resting on. But there's one other catch. What else they found out was that these cells, often when you isolated them from a living organism, they appeared round under the microscope. They had a spherical shape to them. But when you put them in a container, okay, such as a plastic vessel, the cells demonstrated that they were aware of their surrounding environment. They would demonstrate that they were aware when they came in contact with something physical. Okay. And the way they would do this is when they would come in contact with, say, the bottom of what we call a culture dish. Okay. So we put fancy names to these plasticware that we use, call them culture dishes. What would happen is when the cells would come in contact with the material used to make the culture dish, the way the cells would let you know that they're recognizing their surrounding environment is that they would go from a spherical shape, a round shape, to flattening out. They would take on different shapes. Some would have a triangular shape, some would have what we call a spindle shape, some would have what I call a fried egg or over easy shape to them. And what would happen is now if you tried to get them to come off by tilting the dish, tipping it left and right, you would notice they wouldn't move. Well, what was happening is when they would go from a round shape to flattening out, what they were actually doing was producing molecules that would help them attach to the growth surface. So they were actually anchoring themselves. And the only way you could get them off is by chemically treating them to getting them to detach. Then what they would do next, often but not always, if it was cancerous cells that had been isolated and placed in this condition, the cells would grow if you put them in an ideal condition, except for some of these cancerous cell lines, what they would do is one cell would divide into two, and they would grow until you had a single layer of cells covering the growth surface of this culture dish. They would not grow on top of each other. That eventually came to be called contact inhibition. The cells were not only demonstrating they were aware of the growth surface that they were resting on and thus grabbing onto it, but they would also demonstrate that they knew when to stop dividing because they recognized when they came in contact with another cell and how that was different from the growth surface. So they were differentiating between coming in contact with the growth surface and coming in contact with another cell. Well, all this began by taking tissue biopsies. Okay, it could be normal tissue or cancerous tissue. They would disperse or break apart that tissue, and then they would demonstrate how these cells could grow. And then eventually they started taking it further. So in other words, what they were demonstrating is these cells behave much like you and I. Okay, again, let me know if there's too much noise in the background, and I'll move. Okay, next what I want to move into is general cell types. We talked about this in chapter 1. There are two general types of cells. Okay, these two general types of cells are known as prokaryotic cells, such as bacterium, but not only bacterium. Okay, and the other general type of cells, they're known as eukaryotic cells, as we discussed before. Okay, simplest known living organisms, okay, are either prokaryotic cells or eukaryotic cells. The most complex known living organisms, multicellular organisms, they're strictly made up of eukaryotic cells. Now, the obvious difference between prokaryotic and eukaryotic cells is size. You can see here the prokaryotic cell is much, much smaller than a eukaryotic cell, but there are other differences we're going to eventually talk about. Up here in the chart, it tells you the obvious one is prokaryotic cells are smaller, eukaryotic cells are much larger. Okay. Prokaryotic cells are described as having a simpler structure overall, whereas eukaryotic cells overall are described as having a more complex structure. And you're going to see this because we're going to spend a lot more time talking about eukaryotic cells. And these are the cells we're going to focus on mostly, not completely, but mostly throughout the semester. Now, besides the size, as we'll talk about again, another major difference between these two cells has to do with what we call membranous organelles, such as the nucleus. Okay. This is the nucleus of a eukaryotic cell, and you can see the nucleus alone is much larger than the entire prokaryotic cell. Prokaryotic cells don't have a nucleus. However, both these guys contain the compound DNA, which we'll talk about more. Okay, down here it says prokaryotic cells lack most organelles. Well, this is in reference to membranous organelles. By the time we get to the end of the chapter, this should make better sense to you because we will go over types of membranous organelles, but these membranous organelles are only found in eukaryotic cells. There's another group called non-membranous cells, which I eventually will talk about in a sec. Okay, so simplest known living organisms are unicellular. Okay, these unicellular living organisms can be either prokaryotic or eukaryotic. Examples of eukaryotic, well, are kingdom Protista, such as Euglena and Paramecium. You'll see some of these in next week's Lab 4. Okay, those are unicellular eukaryotic living organisms, Euglena and Paramecium. For prokaryotic living organisms, one of the most popular unicellular prokaryotic living organisms is known as Escherichia coli or simply E. coli. Okay, here they're showing you E. coli that is attached to the cells of human tissue. Each of these little rod-shaped things, which you'll come to know as a bacillus shape or a rod shape, but mostly they say bacillus. Each of these is an individual living organism, an individual unicellular prokaryotic living organism known as E. coli. They behave much like you and I. Okay. However, we are multicellular living organisms. Okay. And the way it works for multicellular living organisms is the building blocks that form us are living cells. But not all these living cells are the same. They have differentiated to take on different shapes because they have different functions. That's the purpose of this picture here. In this picture here, the purplish color here is a piece of a skeletal muscle fiber, better known also as a skeletal muscle cell, such as that of the bicep. The green color here is part of another cell type known as a neuron, which is part of the nervous system, specifically the peripheral nervous system. And what they're trying to show you, okay, is that these cell type called neurons, okay, will come right up against and almost in contact with this other type of cell called a skeletal muscle cell or fiber. The reason for this arrangement, okay, is because the skeletal muscle cell will not contract producing movement unless the neuron tells it to do so. All right. So in other words, each of them has a job to do and they're interdependent on each other. Okay. So for multicellular organisms, we are made up of cells that are differentiated, taking on different functions and often have different shapes. But the functions they have in the end should complement each other so that we function as one organism, one individual, just like the E. coli functioning as one individual. Okay, let me move here and I'll move into another one. Sorry about that, everybody. It's just been a little hectic this morning. All right. Hopefully I'm not losing anything here. No problem, Casey. Thank you. All right. Okay, so the next part now. I just went into another room, but not a closet yet. My next move will be to a closet. All right. Contrary to popular belief, everybody, and the administration seems to be lost on this, your faculty have a personal life too. So I apologize about that interruption, but we also have a personal life. Okay, so if you look here, this figure here is to show you, okay, what I mean for multicellular organisms. You take an anatomy and physiology class and A&P class, and they'll go over the various organ systems. One of the organ systems is known as the integumentary system. Okay, it's one of 12 organ systems that forms our body, human body. The integumentary system is most popularly referred to as just the skin, although there's more to it. But when you go over the integumentary system, you talk about the skin, but it's technically known as a cutaneous membrane. It's made up of two different types of tissues, epithelial tissue, which is up here, and connective tissue, which is down here. If you recall, I said there's 4 different types of tissues that contribute to the formation of our body. Therefore, because there's 2 types of tissues, your skin is often referred to as an organ. However, the epithelial tissue, which is made up of many epithelial cells, is technically known as the epidermis. Okay, but if you look here, even with this tissue here, which is epithelial tissue made up of epithelial cells, okay, down here you can see the staining pattern. And these little dark dots represent the nucleus of a cell. You can see the staining pattern here changes as we go from deep inside the skin towards up here, which would be the surface of your skin. Okay. And when they go over this, one thing they should stress, okay, is how your cells, okay, of your body, we're multicellular, how your cells have differentiated to take on special functions. Therefore, the epithelial cells of the epidermis, the reason why they're changing is because they're becoming fully differentiated or highly specialized. And their number one job is to protect everything deep, including this other type of connective tissue known as the dermis. Therefore, when you go over these differentiated cells that contribute to forming different tissues, which in turn contribute to forming different organs and organ systems. Okay, one other thing they'll point out is how they don't have the same shape, but that their shape is often related to their function. So you can see here a cardiac muscle cell of the heart versus a neuron of the nervous system. Okay, they have different shapes, but it's tied into their functions. Now, cells themselves, this is giving you a little scale here. Okay, you've heard about the virus that's going around and people were wearing masks thinking that the mask was going to protect them from contracting the SARS-CoV-2, which is a technical name for the virus that causing the pandemic. Everybody, the virus is down here. Okay, so if you don't have an N95 mask, you're not blocking anything from coming in. The mask they're asking you to wear is actually to protect those around you. It's not going to be protecting you. Okay, but that's why they want everybody to wear one. Okay, so but this figure here is showing you how small cells are. Notice the cells are in the micron range. And if they're prokaryotic cells, they can be down to the nanometer range. Okay, now briefly, okay, I'm not going to go in depth into this. What scientists discovered is cells are small in size to maximize the amount of surface area. Okay, in other words, the surface area referencing the cell or plasma membrane, to maximize the amount of surface area to volume. The volume has to do with what's inside of the cell, the cytoplasm, in order to ensure that the membrane transports, okay, or in order to ensure membrane transport and metabolic needs are met, okay. Us as a multicellular organism, right? We need to breathe, we need to eat, we need the O2, and we need the nutrients, okay? But what actually needs the O2 and the nutrients are the various cells in your body that need it. So your cells eventually will take up those nutrients and O2, which we'll talk about more in chapters 8 and especially 9, okay? But your cells also need to get rid of waste. At a macro scale, okay, at a multicellular living organism scale, we get rid of waste through exhaling CO2. Or in your urine, a lot of people aren't familiar with your urinary system. When you produce urine, that is metabolic waste, mostly made up of metabolic waste that your cells have gotten rid of. And then what you eat, you defecate out. Whatever you don't absorb just passes out the other end. Well, for your cells, they also are taking in what they need, but they're also getting rid of what they don't need. Okay, and in order to ensure that this transport across the cell membrane is occurring at an efficient rate to support metabolic needs, such as the production of energy, okay, ATP, okay, the surface to volume ratio has to be met, okay. Can't have a larger volume than the membrane can support in terms of transport, and we don't need the excess membrane if the volume is at a low amount. That's as far as I'm going to go into it. Usually you'll hear this in your advanced classes, so mainly just remember this statement. If I ask you anything, it'll come off this statement. Okay, briefly, microscopes, because you're going to have a microscope lab. The invention of the microscope, roughly dating back to the 1500s, although it's a person in the 1600s given a lot of credit because they discovered bacteria and protists and things of that nature. I'm not going to go into their names. I'm just going to talk about microscopes. So the microscope, the invention of the microscope allowed for two functions to be performed. The first function is magnification, increasing the size of specimens, in this case cells that were not visible with the naked eye. Okay, but if you wear glasses, you know, if you take your or contacts, if you take your glasses or contacts off, you know, hey, I can make out an object, but that doesn't mean I can't see details. Okay, you may have even walked past somebody not realizing they were a friend of yours and they're telling you hi, and all you see is there's somebody there, but it's a blur. So a microscope performs a second function. In addition to magnification, they also perform a second function called resolution. Okay, resolution is defined technically as the ability to distinguish two entities as separate objects. That's a fancy way of saying being able to see an object, or see the details in an object, or simply see an object clearly. And not only see something's there, I know what it is. I can make it out. That's what we mean by resolution. Now, the most popular scope, the most popular microscope is just simply what we call a light microscope. Some people call it a bright field microscope. It's also known as a compound microscope, which will be discussed in the lab next week. Okay, this is the most popular one, and this is an example of one that you would have used in the lab. Now, there are other types of microscopes that also go under light microscopes. And the reason for this is that whether it's a light or bright field microscope, or whether it's a phase contrast microscope, or whether it's a fluorescent microscope, which I'll show you an image for that in a second, they all rely on a white light source. Okay, here they're showing you a unicellular eukaryotic living organism known as Euglena under a light microscope here. Okay, but there are other types of microscopes that we use because not all living organisms have color to them. This protist here, this single-celled protist here, Euglena, it has organelles called chloroplasts, which give it a green color. So you can see this guy under a light microscope. Okay. However, cells of our body, okay, say the human body, okay, we don't have organelles called chloroplasts. There's no chlorophyll, therefore there's no green color emitted. Okay, our cells are very transparent. Okay, and the only thing you can usually see maybe is the nucleus, and even then you have to stain the cells. Therefore, if you ever end up moving on and you start working with live cells, okay, you'll use a light microscope, but what you'll also use quite a bit is another type of light microscope that has filters in it, creating contrast between the living cells and the surface they're attached to. Okay, these types of scopes are called phase contrast microscopes. They allow you to keep your cells alive, work with your living cells, which are very transparent. So here's one cell. It's a cardiac muscle cell. This little slit here that looks like an eye almost here, that's the nucleus. Everything else around it is a cytoplasm, and then you can barely make out a line, which is a cell membrane. For these cells, these cardiac muscle cells, you wouldn't be able to see that under a light microscope. But phase contrast microscope has filters, okay, to help filter out, in a manner of speaking, the glare and create contrast between the cells and the background to where you can even make out some of the transparent area of the cell. You use these often with live cells, okay, that don't have, say, organelles such as chloroplasts in them. You can also use a white light source, okay, along with fluorescent label dyes, okay, to see specific structures in cells. So we use a white light, but you have these fluorescent filters because you use these fluorescent dyes to help you see particular parts of a cell. Okay, so there are also microscopes called fluorescent microscopes. So often with phase contrast microscope and fluorescent scopes, you won't hear them referred to as light microscopes, but they do have a white light source if you ever use them. You have to turn on the white light, okay? And then you use the additional adapters that they have attached to them. Fluorescent scopes, I used to work with this with students where I worked before with these fluorescent microscopes. And what they would do is we would order from companies fluorescent dyes that emitted a red fluorescent color or a green fluorescent color. And these dyes were chemically attached to molecules that would bind to proteins that made up structures only found in eukaryotic cells called the cytoskeleton. You won't find this structure in prokaryotic cells. Okay, eukaryotic cells were found not to be sacks of jelly. They have a skeleton that they call the cytoskeleton. The cytoskeleton is made up of 3 types of protein fibers. Okay, in this class you'll only be responsible for 2. Therefore, I gave you these figures, and you'll see this again. Okay, 2 of the 3 types of protein fibers that form the cell skeleton or the cytoskeleton are known as microtubules and microfilaments. And what I used to have these students do, okay, is they would perform a fluorescent staining technique, and then they would take their cells on a microscope slide, put it under the fluorescent scope, using the filters, okay, the necessary filters, to help, okay, the red dye fluoresce red. Therefore, what you're seeing here, okay, what you're seeing here, and I'm not trying to outline it, is one cell. This is one cell here. This little dark area here that I'm circling, that is the nucleus of the cell. But everywhere you have the red color, that lets you know where you have these protein fibers that contribute to forming the cell cytoskeleton. It's very extensive. Where I roughly drew the lines, that's where you would find your cell or plasma membrane extending out. And what I would point out to students is that when a cell changes shape and its membrane, say, stretches out like this, we'll notice the microtubules are what are help contributing to extending out the cell membrane. This cell down here has roughly a triangular shape. Here's the nucleus. But then the students would use a different fluorescent dye with a different molecule that would attach to this second type of protein fiber that forms the cytoskeleton. In this case, you can roughly see right here is a cell, okay, and then this is the nucleus of the cell. Okay. But the bottom line was this was to demonstrate to the students how extensive, okay, or how much of these protein fibers you find contributing to the formation of the cytoskeleton, and how the skeleton extends out, also along with the microtubules contributing to the shape here. So we would use fluorescent microscopy. Now there's another type of microscope. Okay, I've never seen one in person. The universities I've been at, you have to have special permission to see these scopes. Okay, they're extremely expensive. Not that the light microscopes we use in the teaching lab are necessarily cheap. They're in the $1,000 range. But these scopes are at a whole nother level. They're called electron microscopes, and they rely on an electron beam to help you see the specimen. Now going back here, this is our protist called the euglena, okay, under a light microscope. Using an electron microscope, we have two different versions of an image of that same organism called euglena. Okay, what it is there were two types of electron microscopes developed or invented. One type is known as the SEM. The S stands for scanning. This type of electron microscope gives you high resolution detail, okay, of the surface of the living organism. So here is that same organism called the euglena, okay, but now you can see we're seeing mainly the cell membrane of it here and how it has striations, and you can see this long little tail looking thing up here, which is known as a flagellum that it has coming out on one end. Now there's another type of electron microscope that was developed known as a TEM. This stands for transmission. It's the TEM microscope that is often used to help study and identify organelles inside of a living cell. This is the same organism, euglena, except now the way they prepare it is they basically have to create slices of the organism, and it takes a trained eye. I couldn't even tell you exactly, other than these are vacuoles, and roughly this is one organelle here and this is one organelle here, but I couldn't tell you what organelle they are. And then if you compare these two images, the SEM versus the TEM, what appears like striations on the surface of this single cell living organism, over here, look at it, it appears as serrated, as a saw. Okay, so but the bottom line is it doesn't matter if it's a light microscope or an electron microscope. Okay, they both perform two functions initially: magnification and resolution. It's just a matter of the level of detail that they give you. Okay, now we want to transition into is back to prokaryotes and eukaryotes, starting with a comparison and then differences. We said for prokaryotic and eukaryotic living organisms, okay, they're actually classified into three domains: domain bacteria, domain archaea, domain eukarya. Now what they all have in common is they all possess the 7 common properties or traits, ranging from evolutionary adaptation to order. Okay, but they're not all the same. Now initially I told you in chapter 1, if a living organism was a prokaryotic living organism, they automatically put them under one kingdom, the kingdom Monera. But with advancements in lab technology, especially DNA sequencing, okay, and identifying genetic information, research scientists discovered, okay, we're wrong. Just because I have 2 unicellular prokaryotic living organisms of different species, okay, but they're prokaryotic, doesn't mean they belong in the same class. Okay, there are differences. We go over this more in 1407 in greater detail, the differences. So for prokaryotic living organisms, what they often have in common is they're unicellular living organisms, okay, but they don't like the same habitats, so therefore they divided them into 2 domains. Okay, eukaryotic living organisms, they're all dumped into one domain. Doesn't matter if they're unicellular or multicellular, they're all dumped into one domain, and then we divide them up. First, what I'm going to start off with is just general traits between prokaryotes and eukaryotes. So in your notes, what I have is when you look at prokaryotes and eukaryotes, besides the obvious, the tremendous difference in size of these 2 general cell types, okay, what they have in common is that they both contain the organic compound, the nucleic acid, DNA, for both of them. Okay. They both contain DNA. The purpose of this is what I said last class, I believe. All living organisms have their genetic information stored in the form of DNA. And you will hear me say this again and again as we go through the semester in both lecture and lab. All living organisms have their genetic information stored in the form of DNA. Therefore, it doesn't matter prokaryotic or eukaryotic living organism, you're going to find DNA. Okay. What else they have in common is that they both have a cell or plasma membrane. In other words, they both have a cell or plasma membrane where the primary organic compound that forms the membrane is a phospholipid. And these phospholipids form a phospholipid bilayer. Okay. What else you have in common that you can't see here between the two is you'll also find the other nucleic acid, RNA, equally important to both, and we'll talk about that later in the semester. And also for both, you will find non-membranous organelles known as ribosomes, which we will talk about ribosomes, but we'll talk about it under eukaryotic cells. Now, you'll find ribosomes in both types of cells because ribosomes are responsible for synthesizing polypeptides. In other words, the cell can't produce a protein to work unless it has ribosomes. It doesn't matter if it's eukaryotic or prokaryotic cell. It needs the ribosomes. Okay, so those are some common features. Now, some differences. Well, if you recall, I said for prokaryotic organisms, whether they're classified under the domain bacteria or the domain archaea, prokaryotic cells tend to exist as unicellular living organisms. I didn't say 100%. I said they tend to exist as unicellular living organisms because most of my students go on to microbiology after this course. Okay? In micro, they'll give you the 100% truth there. But for a large number of prokaryotic living organisms, they exist as unicellular living organisms. Therefore, okay, I'm going to use this other figure here. I have two figures, but they're basically the same. Therefore, for prokaryotic cells, it's not enough to have as an outer barrier protecting everything inside of the cell. It's not enough to have just a cell or plasma membrane. Since they exist as individual living organisms, unicellular prokaryotic living organisms, okay, the cell or plasma membrane is just one of three layers that you find forming the outer barrier of prokaryotic living organisms. The two additional layers are the cell wall and what is called the capsule. Now, just like for the plasma or cell membrane, whereby we have organic compounds called phospholipids forming them, we also have organic compounds contributing to the formation of the cell wall and the capsule. For the cell wall, okay, it depends on the type of prokaryotic living organism. However, at a minimum, the cell wall is made up of proteins and carbohydrates. Now, for some prokaryotes, in addition to proteins and carbohydrates, you will find phospholipids contributing to forming the cell wall. Okay. And then you'll have a third layer known as a capsule. The capsule is primarily formed by polysaccharides, in other words, organic compounds called carbohydrates, but specifically polysaccharides. And the way it works, okay, is the plasma or cell membrane is the inner layer. It's the layer facing what we call the cytoplasm, everything inside here. So you can see the arrow pointing to this color scheme right here. Then wrapped around it, all the way around it, is our next layer called the cell wall. Okay. The purpose of the cell wall, its primary function, protect the organism. This is it. You go into micro, you take a microbiology with a professor such as Felix Villarreal, and you can ask him about antibiotics, and he'll tell you how some antibiotics, this is what the antibiotic destroys, the cell wall, therefore killing off the bacterial infection. And then you have the capsule. Notice they're showing you the capsule forms the outermost layer facing the extracellular or surrounding environment. Okay. The purpose of the capsule is to help the prokaryote stick to a substrate, such as when I showed you the E. coli sticking to human tissue. Okay, it's a capsule that's going to help it attach to human tissue. Now, some prokaryotes, extending out of the capsule, they have protein fibers known as fimbria, but I prefer to use the term pili. So some prokaryotes have these little hair-like projections that extend out of the capsule, and the function of these pili is the same as the capsule. It's to further help the prokaryote attach to a surrounding surface or substrate, except the pili are actually protein fibers extending out of the capsule. Okay, so those are some of the differences, just some of them, between prokaryotes and eukaryotes. Now listen carefully. For eukaryotes, what I want to do first is I want you to go to the end of your notes before I go any further with eukaryotes. There are some terms I want to talk about. And I'm hoping, okay, what's going on? Sorry, this program, I tell you, if it wasn't for other professors telling me it's driving them crazy too, I'd say a lot more. I use more bad words and I wouldn't be able to record this lecture. Okay. This is the last slide, everybody. I want to go over this, and I put it at the end. It's kind of like a glossary, but I put it at the end. Okay. I'm going to use what basically amounts to a terminology that includes directional terminology. It just helps you understand where you are, because as I said, even after the first exam, we continue on with cells, and you're going to hear these terms. So if you look at the last page of your notes, okay, it says terms in reference to cells, or cellular terms, as I sometimes refer to them. The first three terms all end in cellular. Okay, I have extracellular, I have intracellular, and then what I don't have up here is intercellular. Okay, each of them end in cellular, but each of them have a different prefix, and I need to go over this. Some of them, it's usually split down the middle. You know, half my students are familiar with this and they know it. What I tell those students is be thankful. That's one less thing you need to study. But for other students, the other half, we do need to go over this. Okay. Starting with intra, let me start with extra. I'm not going in any particular order, but if you start with extra, extra means outside. Okay, so when we put this prefix in front of cellular, what we're talking about when we say extracellular is we're talking about the extracellular environment, the environment outside of a cell. Okay, this extracellular environment, as we will talk about more towards the end of the chapter, consists of a mixture, okay, sometimes of protein, sugars, and a water-based solution, or sometimes the extracellular environment, there's not much of it because one cell is directly attached to the the membrane of one cell is directly attached to the membrane of another. Okay, but in this case, the way we're going to talk about it is we're going to talk about how you have one cell surrounded by an extracellular environment consisting of proteins, sugars, and a water-based solution. That water-based solution is known as the extracellular fluid. Okay. Except you're not going to hear me say extracellular fluid by chapter 7. What I'm going to say is, if I can do this, I'm doing this with the touchpad. Might actually, well, speaking too soon, I was going to say might actually do better this way. So the abbreviation you're going to hear me use for extracellular fluid is ECF. Okay, going into chapter 7, you'll hear me say ECF. All right. So the extracellular fluid is just part of what forms the extracellular environment, which I use the abbreviation EC. The next two terms, one begins with the prefix intra, and the other one begins with the prefix inter. Do not confuse these two because you are completely wrong if you do. Okay. Intra, I-N-T-R-A, is a prefix that means within or inside, versus extracellular, which means outside. So most of my students will define it as inside in comparison to outside. So if you hear me talk about intracellular environment or intracellular fluid, I'm talking about what's inside of the cell. Okay. Now, inter, the prefix inter means between two or more. So when we use the prefix I-N-T-E-R, intercellular, we're talking about between two or more cells. We will talk about this in chapter 11 when we go over intercellular communication, which I believe the textbook titles cell-to-cell communication or cell communication, but it's how two cells communicate between each other, intercellular. Other terms I have listed to you in no particular order. I said up here the plasma membrane, but I said I also use cell membrane. Okay. When you take your advanced courses, they eventually will focus on plasma. I use both interchangeably. In the case of eukaryotic cells, all eukaryotic cells have a plasma or cell membrane, And the primary compound, just like with prokaryotes, is phospholipids. Now, there is an exception with plants and other organisms, which I'll mention, but we're going to focus mainly on animal cells, which in that case, the outer barrier is only that plasma or cell membrane. This membrane encloses or surrounds the intracellular environment. The intracellular environment is better known as the cytoplasm. Okay? So what I refer to as the intracellular environment, it technically is known as the cytoplasm. The cytoplasm is divided up into two parts. One part are the organelles. You can compare that to, say, if you're inside your home. The organelles would be the furniture. Okay. Surrounding the organelles is a water-based solution called cytosol. You can compare that to the air in your home. Okay. Except what you're going to hear me say by chapter 7, instead of saying cytosol, which is a water-based solution, you're going to hear me use the abbreviation ICF. So in this class, the cytosol is also known as the, excuse me. I got to remind myself, maybe it's the coffee I'm drinking. For some reason, I'm drinking a lot more coffee. I lost count after 3 cups. I know that's a lot. So, again, hopefully y'all are getting a laugh out of this and it keeps your attention. All right. So listen, make sure you make a note. The cytosol, which is a water-based solution, is also known as the ICF. The IC stands for intracellular fluid. Okay. And here at ACC, okay, a lot of our professors, including myself, okay, use ICF because it pairs up better with ECF when we talk about membrane transport, the movement of substances across the membrane, involving an exchange between the ECF and the ICF. Both are water-based solutions, okay? So the cytosol is the ICF is the cytosol. Now, as far as organelles go, notice something. In my day, okay, there was just one group of organelles. Y'all are so lucky that scientists have discovered so much because now they divide organelles into 2 groups. Okay. The 2 groups of organelles are your non-membranous organelles. Some of these non-membranous organelles you also find in prokaryotes, okay, such as ribosomes. Okay. What you don't find in prokaryotes and that you do find only in eukaryotes is this second group of organelles. So when it comes to organelles, the only time we break them up into 2 groups is when we're talking about a eukaryotic cell, because prokaryotic cells do not have membranous organelles. Okay, so for eukaryotic cells, we divide the organelles up into 2 groups: non-membranous organelles, and you can see the list here, and our membranous organelles. Okay, and we're going to go over many of these as we continue on with eukaryotes. All right. So I put it towards the end of the chapter. I was trying to find a place to put it best, but I found it worked better more like a glossary at the end, even though I didn't label it glossary. All right, let's go back now. Okay. I'm briefly going to mention plant cells. Okay, plant cells have a plasma or cell membrane, which they show you here. Except for plant cells, wrapped around that plasma or cell membrane is a cell wall. Except unlike with prokaryotes, for plant cell walls, it's just made up of cellulose, as we talked about in chapter 5. And the main function of the cell wall is to help the plant cell maintain its shape. Okay, some people might say a little bit in water balance, but I just say help it maintain its shape, help with its structure. That's all I'm going to say for plant cells. What I'm going to do, and you're going to see this heavily throughout the semester, is the majority of the time throughout the semester, we are going to focus on animal cells, which are eukaryotic cells. Okay, I have 2 figures here. Each figure has something that the other doesn't. All right. Now, if you look at your notes, okay, anything I skip over, you're still responsible for. I'm just trying to highlight the main points. If you look at your notes, okay, I've given you 2 tables. The first table lists the non-membranous organelles, which you are responsible for. The second table lists the membranous organelles you are responsible for. Some of these organelles we will only mention here, and I may even go over them briefly. Some of these organelles, okay, you will see more of throughout the semester, such as the nucleus, the mitochondria, the rough ER, okay, and the Golgi apparatus. So if you look at your non-membranous organelles, okay, the list of non-membranous organelles, some of them are on this figure here, such as the cytoskeleton, such as microvilli, such as organelles known as centrioles. Okay, this one I'm going to go over because a lot of students that go over what should be centrioles are actually told it's called centrosomes, and no, that's not how it works. The organelles themselves are known as centrioles. So these are a few. Okay, what I did, there's a few extra that I didn't circle on here. Let me find, here's another one, your ribosomes. Okay, what's not on this figure, but it's on the other figure, okay, is another non-membranous organelle, flagellus plural. Flagellum is one. Okay, in future tests, if I don't think we'll be visiting this again. Oh no, we will for cell division. Flagellum, I'm never picky about that. But here's a fifth non-membranous organelle, and this cilia, which is not on any of these figures. Okay, so you have a total of 6 non-membranous organelles, which you're responsible for knowing. I've listed ribosomes last because non-membranous organelles and membranous organelles, okay, are known to work together at times. Ribosomes are the best example to demonstrate that. So I put ribosomes last because that helps me transition into our membranous organelles, beginning with what we call the rough endoplasmic reticulum, or RER. That's why we use abbreviations so we don't choke on our tongue here. All right. So beginning with the cytoskeleton, everybody literally, when I was an undergrad, had a professor describe cells as sacks of jelly, and movement inside was random. Even as an undergrad in his first, second year, okay, I looked at my fellow classmates. We looked at each other and we were like, that doesn't make sense. The sacks of jelly, okay, maybe you're right. Okay, although we should be a little bit, how do you say, less toned. But the random movement absolutely didn't make sense. Because we thought to ourselves, there's no way we would exist, okay, if cells waited for chemical reactions to happen randomly, such as reactions that produce energy needed by a cell to survive and function. Well, as I said, when I was an undergrad, cancer research took off, and that's when it was firmly established that, you know what, first of all, our cells are not sacks of jelly. Our cells have a skeleton, okay, but this is only for eukaryotic cells. They have a skeleton called the cytoskeleton, okay? So, and as I said, the cytoskeleton technically is made up of 3 types of protein fibers, which are all listed here, okay? You're only responsible for 2, but the 3 types are microfilaments, intermediate filaments, and microtubules. I only want you to know 2 for now. It's in advanced courses, and I know some of the professors will add the 3rd one in, but I'm going based off experience. The 3rd group, okay, that contributes to the cytoskeleton, this group, you can get that later in an advanced class. These first 2 are the ones that are most important because you not only hear about them in relation to the cytoskeleton, you hear them in regards to other parts of the cell. So that's why I emphasize these 2. So 2 of the 3 you're responsible for that are protein fibers that form the cytoskeleton are microtubules and microfilaments, okay, and they're pointing to them here, okay, at different sizes. Now, as I said before, where I worked previously, I taught a cell and molecular course, which is a senior level course, and they would do a type of fluorescent microscopy where they would target these 2 out of 3 protein fibers that form the cytoskeleton in animal cells, just to show them how extensive the microtubules are, how they're located running throughout, but not through the nucleus. Same thing for microfilaments, how extensive they are, especially around the cell membrane, but they don't run through the nucleus, which you can see these little dark pill-shaped structures here, okay? So our cells, for eukaryotic cells, they're not sacks of jelly. They have a skeletal system called the cytoskeleton, okay? 2 of the 3 key protein fibers that form them are microtubules and microfilaments. But I also said that professor said movement in a cell is random. Everybody, I can tell you right now, in the eukaryotic cells of our body and in cells, period, random is not movement. I mean, a movement is not random, okay? There's always something promoting movement. And in the case of the cells that form our body, okay, there's definitely not any random movement going on. You take an advanced cell and molecular course, and they will tell you In addition to the first function of the cytoskeleton, which is structural support for the cell, okay, and shape, okay, which we put together, a second function of the cytoskeleton that is equally important is transportation. There are proteins inside of a cell called motor proteins, okay, that will use one of the three protein fibers of the cytoskeleton, microtubules. It will use these microtubules like a rail system or like a roadway. And the motor protein will walk along only the microtubules, transporting substances in a cell intentionally from point A to point B. So if you take an advanced cell molecular course, they go into a lot of detail like I used to go into detail. Okay, I would talk to my students how there are certain molecules that are transported out of the nucleus, okay, into the cytosol. And I would tell them that the molecule that was transported by the motor protein, that molecule had to have a certain ID to get out of the nucleus. Likewise, I would talk about how motor proteins would transport molecules into the nucleus. Those molecules had to have a certain ID in order to gain access into the nucleus. So not only is transportation another function of the cytoskeleton, in other words, there is no random movement, okay? But in addition to that, your cell requires IDs, much the same as if you have a job or if you go to the bank, you have to have the correct ID. So it was anything but sacks of jellies and random. Now, the next three I tend to talk about in a group. These are microvilli, cilia, and flagella. I'm going to use this figure here, okay? If you look structurally speak for all three of them, for microvilli, cilia, and flagellum, they're often described as cytoplasmic extensions, especially cilia and microvilli, not so much with flagellum. If you look here, they're showing you the cell membrane roughly in an orangish color. And if you come over here, notice these finger-like projections, how the membrane is extending up. You can see here where you can see the membrane here, but they cut the cell open so you can see within this little finger-like projection called a cytoplasmic extension. And if you come over here and you look at where it says microfilaments and microtubules, okay, the brown part, which represents the microfilaments, notice those are what are pushing up on the cell membrane, creating these finger-like projections. Whenever that part of the cytoskeleton called microfilaments is pushing up on a cell membrane, creating these short finger-like projections, we call these microvilli. Cells that line your small intestines, okay, and that come in contact with the nutrients passing through your digestive tract, they have microvilli on them. And the reason for having these microvilli is nutrient absorption. Microvilli, by a cell forming microvilli, it allows a cell to increase the amount of surface area, increase the amount of cell membrane by folding it up, but giving it some support with these protein fibers called microfilaments. That way, when nutrients is flowing across, more nutrient comes in contact with more cell membrane, thus allowing the cell to better absorb those nutrients. So that's a primary function for microvilli, and that's the only one I want you to know. However, instead of microfilaments, sometimes microtubules will push up on the cell membrane and create one of the two other structures. Cilia, or as they're showing you here, push out on the cell membrane as a bundle of microtubules, forming a single flagellum. So let me move into these other ones here. So here's just a diagram of tissue, and what they're doing here is they're just created this tissue sample here to show you, hey, here are cells that have microvilli on one side, short hair-like extensions. This is as if you were looking down on cells with microvilli, short hair-like extensions. But over here, they're showing you these are cilia. And here they're showing you cells of the respiratory tract with cilia and microvilli. Okay, so again, microvilli, I just want you to know nutrient absorption, but they have more functions. But here, it's showing you cilia in comparison to microvilli. And here, cilia are also described as hair-like projections, but they're described as long hair-like projections, whereas microvilli are described as short. But what it is cilia are, okay, cytoplasmic extensions. The cell membrane extends out, forming a long finger-like or hair-like projection. What you can't see is it's the microtubules of the cytoskeleton that actually push up on these, giving them some structural support. Now, the reason why I'm telling you this figure here is an actual staying tissue from the respiratory tract is because cilia, their primary function in us is to move substances across the surface of tissues. So for those cells that are what we described as being ciliated, lining our respiratory tract, the substance they're moving across the surface of the tissues of the respiratory tract is mucus. Mucus that is produced by mucus glands. Okay, we need that mucus being produced to protect our cells of our body that are lining the respiratory tract, protect them from infection, protect them from drying out. But we don't want that mucus sitting there, okay, trapping many infectious agents, which eventually can move over. So the cilia will just keep moving mucus along, and new mucus will be moved over it here. So that's a main function. For flagella in animals, there's only one cell that's going to have a flagella, and it's a flagellum, and that's the sperm. Okay. On this figure here, this is the head of the sperm, and the outer barrier is that cell or plasma membrane. Okay, but then that cell or plasma membrane suddenly extends all the way out in the shape of, if you want to call it a tail. But instead of calling it a tail, this is a single flagellum. Okay, so going back to that previous figure, it's hard to find good figures, so please forgive me here. If there's not a good figure, you'll have to look for one, but these are the ones I'm always searching for some. Here they're showing you, it's not connected to the cytoskeleton, but they're showing you here, what they're showing you is a bundle of microtubules that push out the cell membrane. They change the color so it makes it look like this is a separate attachment plugged into the cell membrane. It's not. The cell membrane extends out because the bundle of microtubules push out. Okay. The microtubules, okay, have the ability to move, therefore causing the flagellum to move. Therefore, the function of a flagellum, or the function of flagella in general, is to produce movement of the entire cell, such as a sperm cell. Cause the entire cell to move from point A to point B. So, sorry there's not better figures, okay? These are just some of the ones I've been able to come across here. Now, for the last two non-membranous organelles, okay? Let me see, what do I have here? Let me use these two here, okay? If you look at this figure here, on this figure, they use the term centrioles. I was trying to box that in. Centrioles, okay? And they show you here, they're pointing to these soda can looking things right here. And it says these are centrioles. But if you go to the other figure and you look at it, okay, I might be blind, but to this very day, I've been using this slide for I don't know how many years. I still don't see centrioles on it. Okay, however, I do see this term, centrosomes. And this is pointing to this guys here. Okay, listen carefully, okay? What it is, and I'm going to use this figure. We're going to focus on these guys. These are our next organelles called centrioles, and I'm going to use this other figure because I do have a lot of confusion, and you need to not be confused when we get into chapters 12 and 13, cell division, because we're going to talk about these. What it is there are non-membranous organelles called centrioles. These what I call soda can looking things made up of protein fibers called microtubules. They often depict them as being perpendicular, okay? I haven't found out why, I haven't asked, other than apparently that's how they show up on an electron micrograph, okay? If you notice, the background is highlighted a yellow color. It's this highlighted color that is, referring to centrosomes. So listen carefully. Non-membranous organelles called centrioles are located in an area of the cytoplasm called the centrosome, okay? But the organelles themselves, the non-membranous organelles themselves, are called centrioles. Centrosome is just the region they're located in. As I put in your notes, okay, the function of centrioles, well, they help out with cell division. Specifically, they help with separating what you're going to come to know as sister chromatids into daughter chromosomes when one cell is dividing into two, so that in the end, each cell either has the same number of chromosomes or has the correct number of chromosomes, because there's two different types of cell division, so I have to be careful of what words I'm using here. Now, the last non-membranous organelle, which we are going to use to transition, okay, into our membranous organelles, are our ribosomes. I think I'm going to use this figure here. Hopefully you can see it well, because on my screen kind of looks okay. Using this figure, so again, I have two different figures representing animal cells. One's better than the other in different regards. Ribosomes are our last of 6 types of non-membranous organelles that you're responsible for. Notice something here. The line here pointing to the ribosomes actually branches out. We hit a fork in the road. Here it points to these little beads. But if you notice, some of these little beads, it's showing you up here, are attached to these flattened looking structures that they refer to as flattened membranes. Okay? They're trying to show you is that ribosomes, they always have the same function, no matter where they're located, whether they're out here in the cytosol or whether they are attached to the membrane of a membranous organelle known as the endoplasmic reticulum, or overall better known as the rough ER. It doesn't matter where they're located. The bottom line, the function of a ribosome, initially we say it's to synthesize proteins, and that's how I want you to know going into the test. Later in the semester, we'll be more detailed and say synthesize polypeptides. But going into the test, you can think of it as ribosomes, the function, synthesize proteins. Doesn't matter where they're located. But then the question is, okay, why are they located in 2 different areas within a cell? And that's what we're going to address next. Ribosomes, in general, they're often described as having a function whereby they synthesize proteins. Now, structurally, they are made up of RNA and protein. They're called ribonucleoproteins, and we'll discuss that in detail towards the end of the semester when we go over gene expression. As far as location goes, if the ribosomes are located in the cytosol, and that is where they are synthesizing proteins, we call these ribosomes free ribosomes. Free ribosomes are located in the cytosol, as they show you up here, okay? And they will synthesize proteins that do work inside the cell. In other words, they synthesize proteins that stay in the cell and do their work in the cell, okay? However, sometimes a ribosome synthesizes a protein that even if it stays in the cell, it's going to face some harsh conditions, but often it's not going to stay in the cell. It's either going to be inserted into the cell membrane where part of the protein is facing the extracellular environment, or it is going to be secreted. In other words, it's going to be released into the extracellular environment, which is much harsher, has much harsher conditions than within the cell. Therefore, there are certain proteins that have to do work, okay, in more hazardous environments. Therefore, they have to be modified. They have to be folded in a certain way and treated in a certain way so that they can survive long enough to do their job. In this case, you have ribosomes that are going to attach to membranous organelles called the endoplasmic reticulum. So it's at this point we finish with the first table I gave you, ribosomes, and you turn to the next table. If you look at the next table, it begins with membranous organelles called the endoplasmic reticulum. Except there's 2 types of endoplasmic reticulum. One is a smooth endoplasmic reticulum. The other one we describe as the rough endoplasmic reticulum. Okay, the rough is in reference to ribosomes that are described as fixed ribosomes. This is a second location for ribosomes. Some ribosomes are described as fixed ribosomes, meaning that they are bound or attached to the endoplasmic reticulum, creating the rough endoplasmic reticulum. So notice they're pointing to these here. I'm just using a little bit different terminology here. Okay, now they're still synthesizing proteins, but ribosomes that are fixed ribosomes that are attached to the rough ER, okay? So this is our cell. Here's our plasma or cell membrane. Here's our rough ER over here. Ribosomes that are fixed or that are attached, okay, to the endoplasmic reticulum, thus creating what we call the rough ER, okay? These ribosomes are synthesizing proteins that are immediately passed on to this membranous organelle called the endoplasmic reticulum so that the proteins can be modified, such as being folded in a special way or having sugars added to them. And then what will happen is those proteins will then be transported in a little lipid package called a vesicle, and I'll go over this more again later, transported in a lipid package called a vesicle. And you can't see it, but there's a motor protein walking along the microtubules of the cytoskeleton, taking this little, what is usually spherical in shape, okay, transporting it over to the Golgi apparatus, okay, where these proteins that initially were synthesized by ribosomes of the rough ER are now further modified. And then from there, and they're further modified in an organelle called the Golgi apparatus. I'm going to go over this again, okay, as we go. Golgi apparatus will further modify these proteins, preparing them for the harsher environment they're going to face, okay? So this is why I cover ribosomes last, because we talk about two locations of ribosomes, fixed, okay, versus free. And for the ones that are fixed, this is where you have an example of a non-membranous organelle working hand in hand with the membranous organelle, ER. So I just want to emphasize, okay, they always don't perform functions exclusive of each other. Sometimes they perform functions that complement each other. Okay, let's go back to these cells. I probably need to insert these figures more so I don't go back and forth. Okay, so let me use this other one. I think this other one's a little bit more clean. Okay, so now if you look at the second table, I've listed the membranous organelles that you're responsible for, okay? And basically, you're going to feed off this table. Okay, I might think there's one or two I'm going to give you a little extra that I may not have written any, put it in there that I have students write it in, but you're going to feed off this table heavily. And basically, what I'm talking about is structure and function. But listen carefully. What I do want you to write down is this: membranous organelles. Okay, the way we describe the structure of a membranous organelle. Membranous organelles are formed by enclosed membranes made of the same organic compounds as the cell or plasma membrane. So membranous organelles are formed by enclosed membranes made of the same organic compounds that form your cell membrane, also known as the plasma membrane. Okay? This is not true for non-membranous organelles. So if you look up here, look at the microvilli. Yes, you have cell membrane contributing to the formation of the microvilli, but notice here, it's not enclosed, it's open. It's directly attached to the cytoplasm. But if you look at your membranous organelles, such as your rough ER and your smooth ER, they've cut them open to show you, okay, they are enclosed, but if we cut them open, we can see they have an open cavity or space where something's being stored or a chemical reaction is occurring. Here's the nucleus. Here's the mitochondria. They've also cut those open so you can see, okay, they have membranes that are enclosed and you got to cut them open to see what's on the inside, what's going on. Up here, we have the Golgi apparatus, another membranous organelle. There are peroxisomes and lysosomes, which look like little vesicles that I'll describe later, but they're actually organelles that have particular functions. They're all made up of and formed by an enclosed membrane. And inside of that enclosed membrane is a combination of something being stored and some type of chemical reaction occurring, resulting in the breakdown or the formation of other organic compounds. Okay, it's the best way I can describe membranous organelles. Now, for membranous organelles, we start with the smooth and rough ER. Structurally, they're described as flattened membrane sacs. Okay. Now, the rough ER, the flattened membrane sacs are much more extensive. They're much longer. And the rough has to do with fixed ribosomes that are attached to its membrane. Okay. So it has a dual function. Because the ribosomes are attached to the rough ER gets some credit for that protein synthesis because ribosomes are attached to it. But technically, the ER part of it is, as I said before, modifies the proteins, and any protein synthesized at the rough ER will automatically be transported to the Golgi apparatus. I've never seen an exception to this, okay, in eukaryotic cells, all right? So the function of the rough ER is tied into the function of the Golgi apparatus. And then going backwards, the function of the ribosome is tied into the function of the rough ER. Smooth ER, they're flat membrane sacs, but they're not as extensive, so they show that to you here. However, the function of the smooth ER is equally as important, okay? The function of the smooth ER has to do with the synthesis of lipids. One of the key lipids it's responsible for synthesizing are phospholipids. So if you remember what I said, membranous organelles are formed by an enclosed membrane made up of the same compounds that form the cell membrane. Well, I've already told you that cell, also known as plasma membrane, the key compound that forms it are your phospholipids. So what I am telling you is every membranous organelle that I have listed, its membrane is primarily formed by phospholipids, okay? So this is something we'll talk about also. Therefore, the smooth ER, one of its functions is to synthesize phospholipids, and that is extremely important because a lot of phospholipids are required by the cell. Now, depending on the type of cell, if you take an A&P class, okay, they'll tell you for liver cells, the smooth ER has an additional function besides synthesizing lipids such as phospholipids. The smooth ER of liver cells is also responsible for breaking down toxins that you've ingested when you've consumed food, such as pesticides. If you're talking skeletal muscle cells, if you're talking skeletal muscles made up of skeletal muscle cells, they'll tell you skeletal muscle cells have a smooth ER. In addition to synthesizing phospholipids, they also function as a calcium storage organelle. And as a matter of fact, for skeletal muscle cells, the smooth ER is given a special name, which I'll leave for A&P, just to let you know, okay? So that when you take, if and when you take the class, you know a little bit already of the smooth ER. Now, going back to the rough ER, whatever proteins are synthesized by ribosomes attached to the ER and modified by the ER, those proteins will then be transported to the Golgi apparatus. Okay. The function of the Golgi apparatus is to further modify the proteins. That modification involves attaching sugars or carbohydrates. But a second function of the Golgi apparatus is to sort these proteins. In other words, the Golgi apparatus not only modifies these proteins, but it has to also determine where they're going to be sent. I did not do it. I thought I did. Okay, sorry, but I was looking for a slide I keep forgetting to put in. Let me use this one here. So if you notice here, here's our Golgi apparatus. Never mind the cis and trans. We'll leave that for somebody else. Here's our Golgi apparatus. You can see here's our ER. Even though they don't show you ribosomes here, you can see there's one to the side. Proteins that were synthesized and modified at the rough ER will always be transported in little lipid packages that I call vesicles, or that are called vesicles. They'll be transported to the Golgi apparatus. Okay. Once these proteins arrive at the Golgi apparatus, within the membranes of the Golgi apparatus, they will be further modified by the attachment of carbohydrates and sugars, and they will be sorted. Okay. Some of these proteins will be transported to the cell or plasma membrane where they will be inserted into the membrane. And I'll show you this in chapter 7, a better figure, okay? Where they become membrane proteins that you'll come to know later as transmembrane proteins. Some of these proteins will be sorted and transported to the cell membrane, but they will be secreted or exocytosed outside of the cell. And then some of these proteins that are further modified in the Golgi apparatus will be packaged into what looks like a little lipid package called a vesicle, but in actuality, they're actually being packaged into another type of membranous organelle called a lysosome. Okay. Now, it could be a lysosome, or also it could be another organelle called a peroxisome. Now listen, for peroxisome and vacuoles, I'm not going to go over these. I gave you what I want you to know in the notes. We're not going to visit them later. That's why I'm not going to go in depth right now. That doesn't mean I won't ask you a question, but we don't go in depth. I focus mainly on lysosomes because we mentioned these again. So the way I've tried to list them is to allow us to transition and establish a relationship sometimes for the function of one organelle to another. So one of three directions proteins are sorted from the Golgi apparatus is into organelles called lysosomes. They're packaged into organelles called lysosomes. Lysosomes contain degradative enzymes, sometimes called digestive enzymes, and these enzymes are called acid hydrolases. They're called acid hydrolases because they work very well at an acidic pH. It was found that the environment inside of a lysosome has a pH of 5. Everybody, the cell in general does not want a pH of 5. In general, the cytosol, that ICF solution, has a pH roughly that is neutral, 7.2. Okay, but these enzymes need to do a specific type of work for the cell, and the cell knows this. So what the cell has done is it's evolved. This eukaryotic cell has evolved to where it's packaged these acid hydrolases, these enzymes, okay, into this vesicle, giving them the pH they prefer, a pH of 5, so that they can do their job. Their job is to break down any waste that the cell transports to them, and sometimes that waste might be an old organelle, such as a mitochondria that's not working anymore. So anything that is transported to this organelle called the lysosome will be exposed to acid hydrolases, which are degradative or digestive enzymes, and will be destroyed. Okay. I'll let you look at peroxisomes. It basically follows along that lines. They wanted me to cover it, but to be honest with you, it's best to cover it for advanced classes. Vacuoles, there are different types of vacuoles. Plants have a central vacuole that acts as a water vacuole. Okay, so it helps the plant cell maintain water balance. Sometimes a vacuole might be a food vacuole. Normally, when you hear talk about vacuoles, it's in regards to plants or protists, okay, such as paramecium. I'm going to let y'all look at those. I'm going to jump ahead. Now, I mentioned chloroplast here just because I want you to see this term. Technically speaking, I don't have it on the table, so I'm not going to test you on it. But later in the semester, chapter 9, we're going to talk about the production of energy, chemical energy in the form of ATP. And then in chapter 10, we're going to talk about the production of nutrients. And some of that nutrient serves as chemical energy, such as glucose. So there are two types of membranous organelles. However, one of them you only find in plants. The other one you do find in all eukaryotic living organisms, including plants. They're called mitochondria. Okay, so this will be the one that I'll test you on. This one I'm just mentioning. There's a comparison. Now from there, I'm going to focus on these guys. Mitochondria are the primary sites of a chemical pathway or process known as cellular respiration. The purpose of this chemical process or pathway is to produce a type of energy that the cell needs called ATP. There are many different forms of chemical or potential energy. However, it's like our vehicles. Some of us have vehicles that are hybrids. They run on electricity and gasoline. I believe Tesla's running mainly or only on electricity. My vehicle runs only on gasoline. And then there are vehicles that run on diesel. There are all sources of chemical energy, okay, but not all vehicles can use them. It's the same thing for our cells. Our cells need this type of chemical energy, ATP. Most of this is produced in an organelle called the mitochondria. So the mitochondria is sometimes referred to as a power plant of the cell. Okay, but that I want you to know, be able to tell me it's not only known as a power plant of the cell, but it's the power plant of the cell because it's the site where most ATP is produced. Structurally speaking, the mitochondria, okay, is the only organelle in animal cells, okay, and one of two organelles in plant cells that has a double membrane. Okay, actually the nucleus does too, but I put the nucleus in the middle because the mitochondria, its membranes are separated by a space. They don't come in contact with each other. Okay, within the inner membrane, it was discovered mitochondria have their own ribosomes. Furthermore, it was discovered that in eukaryotic cells, it ends up you just don't find DNA in the nucleus of the cell. In eukaryotic cells, the mitochondria have their own DNA, and it does contain genetic information. You may have heard this before. Okay, maternal mitochondrial DNA. Some companies will use it to establish a maternal lineage relationship from one generation to the other. That's because the majority of the mitochondria inherited by offspring, or usually the mitochondria inherited by offspring, come from mom. Okay. In biology, I try never to say 100%. I was taught that in grad school, and I found it to always be true. Life loves to throw you curveballs. But this is some of the information here. We'll go over this organelle in more detail, but for now, know what I gave you in the notes. That's the main thing. Then there's the nucleus. We're going to visit this a lot. Okay, so it's very important you not only know the anatomy going into the test, you need to know it later in the semester. That's why I said, okay, the exam on Tuesday, it is not a take-home exam. It is not an open notes exam. You need to study for it, okay, and then take the exam. It's multiple choice, but more importantly, you need this information for later in the semester, and I don't guarantee you that future exams will be multiple choice. So please, you're investing in the future here too, not just your exam, okay? All right, so if you look here, the nucleus has a membrane, and unlike most, if not all, the other membranous organelles, the membrane of the nucleus is given a special name. It's called the nuclear envelope. You will hear me use this a lot later, okay? The nuclear envelope, okay, is the membrane of the nucleus, and it's referred to as consisting of a double layer, an inner and outer. But the reason why I say it's different from the mitochondrial membrane is because there are points where the membrane, the inner and outer, actually connect, come in contact. You don't see that with the mitochondria. The inner and outer membrane, they're separate from each other. There's a slight space. So there's a little bit of differentiation. Now, why is it for the nucleus, which has a membrane called the nuclear envelope, okay, which is said to have an inner and outer, why would these two membranes come in contact ever? Well, because throughout the nuclear envelope, you have proteins which form openings into the nucleus called nuclear pores. These nuclear pores are used as doorways, okay, but the door for some molecules that are being transported, okay, only works exiting the nucleus. The doorway for other molecules only works for molecules that are entering the nucleus. And then for other molecules that are being transported, they can be transported to and from the nucleus through these nuclear pores. So the pore serves as a doorway, but that doesn't mean any molecule has free access, okay? Now, I believe most of my students are familiar with the nucleus is most popularly known for storing DNA. But if you look at this figure, nowhere on this figure does it say DNA, okay? So I'm going to let you know. Just like I highlighted nuclear envelope, just like I highlighted nuclear pore, just like I highlighted nucleus, notice I highlighted chromatin. Everybody, whenever you see the terms chromatin, chromosome, sister chromatids, whenever you hear any of those three terms, automatically tell yourself they are talking about DNA. So instead of saying DNA, they say chromatin, but chromatin is in reference to how the DNA is packed within the nucleus. It's loosely packed within the nucleus. Therefore, it is definitely taking up all the space or most of the space in the nucleus. So this is whenever DNA is loosely packed, we call it chromatin. So this is DNA. So make a little note for yourself where it says chromatin, they're talking DNA. Now, what scientists notice is that whenever they stain a cell, there's a region of the nucleus within the nucleus, inside of it, that tends to stain more intensely, okay, stain darker than the rest of the nucleus, okay? Listen carefully. This is not a separate organelle. This is a region, an area within the nucleus. They call it the nucleolus. What they found out is when they use general dyes, general stains or dyes to stain the nucleus of a cell, these general stains, okay, bind to organic compounds. Therefore, had we met in person, we would have done a cheek cell stain. And a lot of my students think that the cell itself is this little bean-shaped structure under the microscope. And I have to tell them, no, that's the nucleus. You have to adjust the scope so you can see the rest of the cytoplasm in the cell membrane. Okay, but what will happen sometimes is you'll get a much more intense stained area within the nucleus. What they found out is that ribosomes that synthesize proteins, they're actually put together here. They're assembled here within the nucleus. So what we have now is not only DNA in the form of chromatin, but we have our RNA and proteins that are being put together to form ribosomes. Therefore, you have a much higher concentration of organic compounds here. Therefore, it stains darker when you use a general stain, so they started calling this a nucleolus. But it's where you not only have DNA in the form of chromatin, but it's where ribosomes will be assembled. All right, let's transition into our cell or plasma membrane. Okay, I tend to use both. For the test, I'm going to use both for y'all online, often in class, just to avoid tongue twisting. I say cell. If you look at this figure, look at your notes. Okay, the way I start off talking about the cell membrane is relating it to chapter 5. Yeah, only in the nucleus. Yeah, so the question is chromatin only referred to as such when it is in the nucleus? Yeah, often that's the way we refer to it. So when we go over cell division, that's when we'll add in the terms chromosome and sister chromatids, because the only time you see DNA forming a chromosome or sister chromatids is when it's dividing. When a cell is at rest, that the DNA is in the nucleus and only the nucleus, it exists as chromatin. So thank you, Casey. That was a good question. Thank you. Okay, for the cell membrane now. Okay, this outer barrier, also known as the plasma membrane. If you look, what I did is I initially listed the organic compounds we need to form it. Now, did I list all of them? No. There are carbohydrates, which they're showing you here, but I'm not going to talk about those because I'm only going to focus on the compounds that we need for chapter 7 when we talk about how the membrane functions specifically. Okay, so if you look at the compounds I listed, the key compounds we need, the main organic compounds we need to put together the plasma or cell membrane are lipids and proteins. Now, there are 4 types of lipids. We only need 2. Okay, we need our phospholipids, if you remember the little head and tails or ball and chain, and we need cholesterol, which they show you here. But in addition to that, we need our second group of compounds. We need our proteins. Okay, and if you look there for proteins, you have 3 different types of proteins which contribute to forming the cell membrane. However, it's the first type I've listed. In your notes, I use the simplest term for these proteins. They're called transmembrane proteins. Trans means through. These are proteins that are part of the cell membrane. They run through the membrane to where part of the protein forms the extracellular environment or fluid, and part of the protein forms the cytoplasm, cytosol, or ICF. Okay? Listen carefully, make a note. If you take an advanced class, they won't say transmembrane protein. They will say integral protein. An integral protein, which is here, is a transmembrane protein. It's one of these. But notice for the transmembrane proteins, they're showing you they have different shapes. Okay, and I'll discuss that more in chapter 7 after the first exam. Now, there are other proteins, such as this protein here. It's not actually embedded or inserted into the membrane. It's attached to one of these transmembrane proteins. It's a peripheral protein. When we get to chapter 7, I'll show you what I mean by lipid anchors. So right now, you just need to know lipids and proteins. And for proteins, just know the 3 types. And this will be the main one we'll focus on when we talk about membrane transport. Now, for the lipids, as I said, there's phospholipids and there's cholesterol. Now, keep in mind, these 2 compounds that form the plasma or cell membrane, these are the same compounds that form the membrane of your membranous organelle. So if we were to take the nuclear envelope of the nucleus and dissect it into the organic compounds that form the nuclear envelope, they should be synonymous or mostly synonymous with the same compounds that make up the cell membrane. Okay, I need y'all to remember that for future reference because there'll be some processes I'm talking about that you need to know this. So we already said before, phospholipids are the major compound. They not only are the major compound that form the majority of the cell or plasma membrane, but they form a bilayer. Okay, and I already talked before what the phospholipids are made up of and how these 2 layers of phospholipids, the fatty acids, face each other, creating a hydrophobic barrier. It says tails, but barrier here. Okay, and how the polar head groups face either the ECF in the extracellular environment or the ICF, known as the solution in the ICF or cytosol over here. Now, other compounds that contribute to forming the cell membrane, as I said before, are cholesterol, which is back on this figure here. You can see, though, with cholesterol, it looks like little pegs inserted into the membrane, and they don't run the length of the membrane. You can either see cholesterol here on this side of the membrane or cholesterol here. Now, I'm not going to test you on this, but in advanced classes, what they will do is instead of continuously referring to this as a phospholipid bilayer, they will start referring to this part of the phospholipid bilayer as the inner leaflet and this part of the phospholipid bilayer as the outer leaflet. I'm only referring to it as the phospholipid bilayer, but you can see the cholesterol's in one layer or the other, and then the transmembrane proteins run the length. Now, if you look, I give you 4 functions for the cell membrane. Okay, the number 1 function is it acts as a physical barrier because in the extracellular environment, we have a water-based solution made up of solutes and the solvent water that we call extracellular fluid or ECF. Inside of the cell, we have the cytoplasm, and part of the cytoplasm is made up of a water-based solution known as a cytosol or simply ICF. So by phospholipids being the primary compounds that form the cell membrane, and by the fatty acids of each layer facing each other, this creates a hydrophobic barrier, which makes it hard for the majority of ions and molecules to cross this barrier. Therefore, the number 1 function of the cell membrane, physical barrier. However, if you look, not all these transmembrane proteins, okay, have the same shape. Okay, and this is where the other 3 traits are, which I'm only going to mention a couple. Okay. These proteins and even the phospholipid bilayer, okay, also contributing to regulating what can cross the membrane. So we'll talk about in chapter 7 that this hydrophobic barrier created by the phospholipid bilayer, it's not impermeable, it's semi-permeable. We'll talk about how lipids and gases can pass through it, but most molecules that are water soluble or dissolve in water, as well as ions, can't get through it. Thus, we have transmembrane proteins. Some transmembrane proteins, if you look up here, function as channel proteins. This is to help ensure the cell does take in or get rid of what it needs to if it can't get across this barrier. So transport a nutrient, transport a waste products, but then you have proteins such as this and this here. You have other transmembrane proteins, and they're responsible for detecting chemical cues in the surrounding environment, or maybe their job is to attach to surrounding proteins and sugars in the extracellular environment, which we don't call extracellular fluid. Instead, we call it our extracellular, let me jump ahead and then I'm going to jump back. Instead, we call it our extracellular matrix. So I'm going to jump around a little bit here. This is not true for all cells that form all tissues, but it definitely for some several types of connective tissues. Okay, with connective tissues, often what you see are cells that are not up against each other. They're separated by some space, and filling that space is extracellular fluid and proteins and sugars that form the extracellular matrix. So sometimes a transmembrane protein in the cell membrane will attach to this matrix, helping anchor the cell down, which is what this figure is showing you. Here's the plasma membrane or cell membrane with the phospholipid bilayer. Here's our transmembrane proteins, except they're attaching to the protein part of our extracellular environment called the extracellular matrix, which is surrounded by fluid. Okay, so we call these guys CAMs, but more specifically in this case, if these transmembrane proteins are attaching the cell membrane to this extracellular matrix of the extracellular environment, we don't just call them CAMs, we call them integrins. Now, CAM stands for cell adhesion molecules. So I jumped out of line here, but going back now, I'm on your last page now, everybody. Okay. We have a class of proteins known as CAMs. That CAM stands for cell adhesion molecules. These are transmembrane proteins that help anchor the membrane of one cell to the membrane of another cell or to the extracellular matrix of the extracellular environment. Whenever CAMs in the cell membrane of one cell bind to CAMs in the cell membrane of another cell, a junction is created or an intersection. Okay, you find this frequently with epithelial cells. I'm sorry, with epithelial tissue and muscle tissue, where the cells are up against each other, but especially with epithelial tissue made up of epithelial cells, you will definitely find cell junctions. Okay, so here they're showing you there are 4 types of not just cell junctions, but notice intercellular junctions, where the membranes of 2 separate cells not only come in contact, but they're actually attached to each other. If it involves plants, we call that cell junction a plasmodesmata. Okay. So they're not showing you in great detail, but you can see here there's little lines right here and here. The opening is called the plasmodesmata, but what you can't see is you have proteins called CAMs, okay, that when they attach to each other, and also, are inserted into the cell wall as well as the cell membrane, but when they attach to each other, they create a little opening or doorway between these 2 cells. Now, what that does is it allows what's in the cytoplasm of one plant cell to easily exchange with what's in the cytoplasm of the other cell if the substance is small enough to pass through. Now, you don't hear much for plasmodesmatas. Instead, what you hear, is talk about gap junctions. I'm going to start with gap junctions and then go backwards, okay? Gap junctions are most popularly talked about when you go over the heart muscles, cardiac muscle tissue, okay? But if you look down here, there are cell adhesion molecules called CAMs, which I put here in the blue box, okay, that you will find in the membranes of 2 different cells that are up against each other. And these CAMs will attach to each other, creating a little pore or doorway denoted by this red line. This will allow the cells to exchange, exchange substances between each other. When you go over the cardiovascular system and they talk about gap junctions between your cardiac muscle cells, in that case, the gap junctions aren't necessarily involved in this exchange of substances per se, but they are exchanged in ion exchange, which allows an electrical charge to move along the membrane. I know it doesn't make much sense right now, but just remember that if you take an A&P class, and they'll go over it. Now, there are other types of junctions you find between animal cells. Okay, sometimes you will have cell adhesion molecules called CAMs attaching the membrane of 2 separate cells, forming what we call tight junctions. This you commonly see along the digestive tract. Okay, and one of the most popular sites they will talk about these tight junctions is along the small intestines. They will not only talk about microvilli, okay, on cells lining the digestive tract of the small intestines, but they will talk about how the membranes of neighboring cells are tightly attached to each other through cams throughout. Almost looks like somebody put a sewing machine to this and stitched them up. That way, as food is passing across the surface here, the microvilli are doing their job of nutrient absorption, but should anything try to squeeze between the cells to get access to your deep tissues, it's rejected because of these tight junctions. Then there are cams that form desmosomes, and often you hear these referred to as button desmosomes. It just randomly attaches the membrane of one cell to another to help contribute to structural stability between the two cells. Now, if you look at these, I keep saying these are cells. They don't look like cells. Well, what they've done is they've cleaned out the cytoplasm. They've taken the cytosol and all the organelles out so that we're looking along the inner surface of this cell membrane and the inner surface of this cell membrane so that they can make these proteins called CAMs stand out where they are and how they form various cell junctions, such as tight junctions, which are continuous, such as desmosomes, which are non-continuous. You can see they're just in specific areas, or so they can show you how CAMs can form what we call gap junctions, okay, which are not continuous, but they create little pores connecting the cytosol between two cells to each other. Okay. Are there any questions right now? I know it's a lot of information, but like I said, study, study, study. Listen, the advantage is this. I've never given just a multiple choice question for the first exam. This is a first. Okay, but I don't want anybody to take it for granted. If you study, if you do your due diligence and you study and you complete your work and what have not and go over the reviews, you should be in a good position not only to do well in the test, but actually, to me, more importantly, you're ready to go for the next chapters that we're going to cover. This course builds on itself, everybody, okay? And especially if you aspire to go into the allied health field or any program, really, hopefully you understand the importance of not just getting a good test grade and getting a good final grade, but learning the information. So again, are there any questions? No, that's this figure right here. Okay, so no, that's fine. That's why I'm asking. So, no, that's a good question. Okay, I was talking to one of my family members saying that maybe I should get one of my young ones here that was making noise. Every once in a while, put them on the mic to scream a little to get everybody to wake up. So it's okay. No, it's a good question. So here we have the cytoplasm, and they're showing you here how it's divided up into two parts. Okay, one of those parts is your cytosol, and hopefully, how ugly I'm drawing it will kind of leave a good imprint in your mind. Does that help you out there, Casey? No. I gave you the review and the PowerPoints, and the test's going to come off of what I talked about. Rely heavily on your notes, Edgar. You're welcome, Casey. And just listen to these lectures, okay? Okay, if there are no further questions, I'm going to end the session. But please remember, okay, I pre-recorded the lab, so it's up to y'all to listen to the lab. Lab 3 lab report is due next Tuesday at noon. Okay, so make sure when you click on the link, it'll show you the due date. But remember, Lab 2, you have till 10 p.m. and I'm going to try to remind me to do that for Tuesday's lab to give y'all till the evening to turn in whatever lab's assigned on Tuesday. Make sure I give you till 10 p.m. on Thursday. Okay, but the lab you have assigned on Thursday, you got more than enough time. Okay, y'all take care, stay safe, study hard, please. Okay, bye.